In problem 1518, the question asks, what genetic defects result in the disorder xeroderma pigmentosum in humans? How do these defects create the phenotypes associated with the disorder? So for this, we can go back to our lecture on October 28th, right? So we were talking about mutations, and we were talking a little bit about UV light. So UV light happens to, okay, so there are photons associated with UV light, and they have a certain level of energy associated with them. And one type of damage that UV light causes is this damage that we like to call pyrimidine dimers. So that doesn't look right. Pyrimidine, okay, that's right. Pyrimidine dimers. Okay, so what is this? And normally pyrimidine dimers are forming between two thymine bases. So if we have, okay, let me, I'll diagram a strand of DNA here, starting with, let's see, cytosine, why not? A, T, T, I probably put too many bases in there before I got to the T's, but let's see if how this works out. So 5' end, 3' end, and 3' end over here. So G, C, T, T, A, C, T, and G. Okay. Now, okay, we can put our hydrogen bonds in here. Okay, 2 between the A's and T's, 3 between the G's and C's. Okay, so what is going to happen here with UV light? Okay, so we go out in the sun, we're at the beach, you get a sunburn, or I try to avoid the sun as much as possible, but you know, my family, my kids want to go to the beach, so okay, so I'm out there once in a while, and they're out there, and we wear our sunscreen, but you know, it's not perfect. And so the UV light gets through, and so we have, you know, in our cells we have DNA, and so when that UV light hits the DNA, or those photons, if it hits it perfectly, it has a certain amount of energy which can cause these covalent bonds to form between the adjacent thymine bases. Okay, and so what is that going to do? Well, let me erase this right here. Erase those hydrogen bonds here. And the way we like to diagram this is we put the T's out here. Where's my pen? Pen. Okay. So now the T's are out here, right? Okay, and I don't even know really what's going on here. I don't think that the hydrogen bonding, I think it's all screwed up over there. Okay, but we have our pyrimidine dimers, so we get a bunch of bonds now. These are formed actually between the adjacent T's, so formed between the nitrogenous bases that are next to each other. We never see that, right? We never see that in our structure of DNA. Covalent bonds, not hydrogen bonds, strong bonds being formed between the bases. And that screws up, you know, that really messes up the structure of DNA. So it's supposed to be like a winding double helix, right? And everything's perfect, everything's perfectly spaced from one another. And then we put those bonds in there, and okay, so we get what is called a bulky lesion. Okay, so when we go out in the sun, we get these things everywhere. Actually, don't know how many we get, but as we can tell, if you have xeroderma pigmentosum and you can't fix these, well, terrible things happen. So for those of us who don't have xeroderma pigmentosum, we can tell that our DNA repair machinery is doing a lot of work to fix all these things. So, and how does that work? Okay, so we have a DNA, we have many different DNA repair processes, but the one that is mostly involved with fixing this sort of damage right here, and we need to fix this, right? Because let's say this goes through replication, when this strand right here on the right, if that was going to be used for replication, if this was going to be the template, well then when we got to this lesion part, well you can probably see that, or guess that the DNA polymerase would have trouble using this as a template, and it might actually might skip this area, jump over it, it might put in the wrong base. The hydrogen bonding is all screwed up, we think, between here, so it doesn't know what base to put in, so it can lead to mutations. So we need to fix these, or the cell needs to fix those. We don't have to do anything. Isn't that great? We can just go out in the sun and our cells will fix most of the damage. Okay, but the process we think that is mostly involved in fixing these is called nucleotide excision repair. So nucleotide excision repair, let me write that out. Nucleotide excision repair. Okay, and so how does this process work? We don't need to know the details for our class this year of how this process works. We'll cover some of these in another plug for BSc 350. For those of you who are thinking of advanced graduate work in molecular biology, then this is a class you'd want to take. So, but how for now, for answering this question, how does this work? Well, essentially with NER, what happens is there are proteins which recognize this damage here, proteins which recognize the damage, and they recruit other proteins here to the damage, and what it does is it cuts a specific number of bases out on either side of this damage. I think it's like 10 or 20 or something like that. So it cuts out a big segment of this DNA right here. Okay, cuts it out, gets rid of it, disposes of it, and then your polymerases and your ligases come in, and what they do is they just copy everything. So let me cut that out with the eraser here. I need a bigger eraser. Alright, okay. Get rid of all that. No, that's not working. How about this? I think you know where I'm going with this anyways, right? So cut it out and just fill it back in using the this guy over here as the template. And okay, and we have our repaired DNA. Okay, perfect. So that's one of the repair processes that, you know, this fixes other things other than pyrimidine dimers or thymine dimers. So it's a very important repair process, but it happens to also be very important for fixing those thymine dimers that result from going out in the sun. So let's see. Let's go back to the question now. Looking at... Okay, yep. So what genetic defects result in the disorder xeroderma pigmentosum? Okay, so XP, if you have this genetic disorder, you usually have a mutation in in a gene required for nucleotide excision repair, and we're assuming all of these mutations are recessive, so you carry 2 mutant copies of a gene. Let's call one gene XP1. So you have one XP1 mutant you inherited one from your mom, and you inherited one from your dad, and now you have xeroderma pigmentosum. And there are a whole number of genes involved in this process, in addition to your standard DNA polymerase and your ligase that are required. Other proteins that are involved with recognizing the damage and recruiting all the repair processes there and cutting out the DNA, a specific number of bases or nucleotides. So a number of different processes, and you could be homozygous recessive for any number of those mutant genes and result in having the genetic disorder. in the last part of this question, how do these defects create the phenotypes associated with the disorder? Well, we kind of already went over that, right? So you have the damage, you can't fix it, so you end up with a disorder. You end up with a bunch of mutations that can't be repaired after exposure to UV light in the sun, from the sun, right? The sun has UV rays and visible light, and if you have XP, you can't fix the damage to your DNA resulting from that sunlight. Okay, so that's the answer to 518, and we'll talk more about XP and some of the later problems too.